Hey, Mr. B here. In this video, I am following up my two prior videos that were titled Digestive System and Enzymatic Activity related to the digestive tract with this video, which is all about the absorption of the nutrients that are basically produced via digestion. So far in the previous videos, we've talked about how the digestive system is set up, all of the structural components of the digestive system. We've talked about what the roles of all of those digestive system components are. We also talked about enzymatic activity. We talked a little bit about hormonal control. And so all of that is foundational to how the body actually absorbs the nutrients that have been digested from the polymers that were in the food that you consumed through ingestion. Okay, so for the purposes of this video, we're just focusing on the absorption the portion of digestion and absorption, and so let's get started. So we've talked in depth in previous videos about what the role of the particular organs are holistically within the digestive system, but now we're going to focus on specifically the ilium. Okay, because if you look at this particular diagram, we've already talked that the duodenum is kind of this upper portion of the digestive tract. There is technically another kind of middle section called the jejunum. We don't necessarily need to worry about it. The duodenum and ilium are kind of, for IB purposes, the most two important parts. But the ilium, this back half, or the back part of the digestive system in terms of the small intestine at least, is where the absorption takes place. Okay, so we are focusing our discussion in this video specifically to the ilium, which has a role that is specifically designed to absorb the nutrients that have been digested from the polymers that consumed as food. Okay, so if we kind of zoom into these structures, you will notice that the tube that actually makes up the small intestines has two layers of muscle. Okay, we've talked about it in a previous video. You know that there is longitudinal muscle, and you know that there is circular muscle. Okay, and these circular muscles layer is inside of the longitudinal layer. But if we zoom in even farther, you will notice that in this kind of mucosa layer, which is on the inside of the actual circulatory muscle layer, there is actually these tiny projections called villi, which when we zoom in actually look like tiny finger projections that extend into the lumen, which is greatly increasing the amount of surface area the inside of this tube actually has in terms of its absorption availability or absorption efficiency. And it's these villi that have all of these individual villus or microvilli attached that increase surface area enough to actually make absorption a viable, really efficient process, and one that digestion is really dependent on, right? And so for the purposes of this particular slide, I think that all you need to know really is that as you zoom in to the mucosa layer, which is kind of this peachy pinky layer that is on the direct inside touching the lumen, you have all of these villus or all of these villi that are completely surrounded with these microvilli that are all kind of synergistically working together to increase surface area so that we can absorb as many nutrients as possible from the food that has been digested on its way to the ilium. Okay? When we look at transverse sections of small intestine with either a micrograph or a microscope, you will see this particular structure, and this is an actual skill. Okay? This is outlined in the IB subject guide as a skill that is required for you to know. And so when you look at this particular tissue layer, we need to be able to identify, or when you look at this cross section or this transverse section, we need to be able to identify particular or specific tissue layers. And so if we work from outside in, you will see that there is on the outside a layer called the serosa. Moving inside of the serosa, you will see that there is actually this muscular layer, which is made up of kind of both longitudinal on the outside and circular on the inside. There would technically be a division somewhere in the middle. It's really hard to tell. But this pinky color that kind of looks like a border around this entire transverse section is the muscular layer. The muscular layer, remember, is made up of longitudinal and circular smooth muscle controlled by your ANS. Inside of the muscular layer, you will see another layer, which is the submucosa. Inside of the submucosa, you will see that there is actually that mucosa layer, which is the layer that contains the villus or the villi, which can be seen as these individual projections that are extending into the lumen, which I didn't label, but that would be this space. Okay, lumen is the inside space of the tube, and it is the space that the food contents and the digestive matter is moving through. Now, by incorporating all of these tiny projections, you can see how much surface area those tubes actually have and why they are so efficient at absorbing nutrients. Okay, so what do these intestinal villi look like? If we kind of take off all of the the epithelial layer or the epithelial cells and we kind of look through this structure, you will see a couple different structures. Obviously, there is a single cell thick skin layer, which is important because we have short diffusion paths. The epithelial cell that is made up of this particular tissue looks like this. This is what the actual microvilli projections look like when you zoom in. Okay, and so every single one of these epithelial cells would have all of these individual microvilli, which just further increases surface area in addition to the villus or these villi, which are the larger projections that you see on the actual micrograph. Inside of this villus, you will notice that there is a network of capillaries, both blue and red, which basically is either the artery or the vein. And I know that this is such a small structure, so I'm aware that it is not a artery or a vein, but it is an arteriole because it is supplying artery blood, which is oxygen-rich blood, to the villus. The venule is the vein-related blood, which is the deoxygenated blood. It also is carbon dioxide rich because it is coming from the cells, and so blood is coming in through the arteriole, blood is leaving through the venule in these particular villus structures. Now, there also is lymphatic tissue, or the lymph vessel, that comes into this particular villus as well, and it runs right through the middle of all of the blood supply, okay, the network of capillaries, and this lacteole, which is the name of this structure, has an important role, which we'll talk about in a minute, okay? So, the selective absorbing molecules found in the lumen of the small intestine are going to be absorbed by the epithelial cells through the microvilli and are only going to have to diffuse through one cell thick worth of tissue in order to get into the blood supply associated with this capillary network. Microvilli greatly increase the surface area for absorption and extend into the lumen of the intestine even more than the villus extends into the particular lumen of the intestine. Most monomers are absorbed into the capillary bed. Okay, obviously these blood capillaries make up this network of capillaries, which we refer to as the capillary bed. However, some of the larger monomers, such as fatty acids, are absorbed first into the lacteole. Okay, So, some of the monomers that are going to be easily absorbed by either the capillary bed or the lacteal are all included in this list. Water is going to be absorbed, glucose, amino acids, nucleotides, glycerol, fatty acids, mineral ions, and vitamins. Glucose to fatty acids are all monomers that have been discussed in previous videos, right? In previous topics associated with this class, we have studied in depth this group of organic molecules. Water obviously is not an organic molecule, it's just H2O, but is definitely important when we talk about life. Mineral ions and vitamins are not organic either, but they are definitely a part of food, and they are definitely a part of the digestive matter and are essential for us to absorb from the food we eat. Okay, a lot of these things can actually be absorbed directly by the capillary bed, but some, if they're too big, such as fatty acids, and that is kind of the most important one that is absorbed by the lacteal, absorbed by the lacteal and not associated with the capillary bed. Okay, what are the transport mechanisms that are at play in the absorption of nutrients from the process called digestion? We have, just like in other places of the IB biology syllabus, or just any different areas of biology even, you have passive transport mechanisms and active transport mechanisms. Okay, it's no different. A lot of these kind of bigger biological processes really rely on some of the foundational processes that we've talked about throughout the syllabus. When we talk passive transport, we obviously have to talk about diffusion and facilitated diffusion. Diffusion is when materials that are either very small or non-polar and sometimes even non-charged, they can just easily diffuse through this membrane following their natural concentration gradient. So if they're following a high to low concentration gradient, and they are nonpolar, or they are really, really small, or they are uncharged, they likely can just fit directly through the membrane, and it's just called diffusion. If the molecules are slightly bigger, such as glucose or amino acids, they often have to travel through channel proteins or transport proteins, using a process or utilizing a process called facilitated diffusion. Okay, facilitated diffusion is diffusion. They're still following their natural concentration gradient from high to low, but they are often too big to move directly through the membrane, or they're polar, or they're charged, and so they can't fit directly through the membrane. They must travel a specifically designed channel in order to get across the membrane. Okay? So, things like glucose and amino acids have to travel through a specific channel protein or a particular carrier protein in order to successfully pass through the membrane due to a lot of times their size or polarity. In the event we need to actively pump a molecule against the concentration gradient, we employ an active transport mechanism, which in some cases or under certain circumstances is glucose and amino acids. They have to actively be pumped, and most of the time they use ATP energy in order to get them from an area of low to an area of high concentration. Okay, notice the arrow and the solutes that we're following. This is the low side for these particular square solutes. They're being pumped to the high side using energy against their concentration gradient. That would be a form of active transport. Okay, endocytosis, which is both pinocytosis and phagocytosis, both are a form of bulk transport and require ATP expenditure in order to move materials that are very, very large into the cell, utilizing a membrane in the form of a vesicle. Certain materials that are moved in or out of a cell via bulk transport require an ATP expenditure, and due to the enormous size of material that is being transported, that belongs to the active transport mechanism as well. Okay? If you have any questions about the absorption specific process, make sure you review some of the slides in this video or bring your questions to class. I will see you later. Bye.